Let's talk about gene cloning. Now, I know it sounds like something straight out of a sci-fi movie, but it's actually one of the most fundamental tools in all of modern biology. At its heart, it's really about one thing: finding a single tiny piece of genetic code and making millions or even billions of copies. It's like building a factory for a single gene. But how on earth do you even get started?
Well, imagine this. I tell you to go into a library with millions of books, find one very specific sentence, and then print a billion perfect copies of it. That sounds impossible, right? But that's pretty much the scale of the problem scientists are up against. A cell's DNA is that massive library, and a single gene is that one single sentence. To even begin to pull this off, they have to solve two huge problems.
First up is amplification. How do you make enough copies of that one sentence so you can actually study it or use it? And second is purification. You know, in the messy soup of DNA that you create in a lab, how do you find and pull out the one correct molecule you're looking for and get rid of all the other junk? These two challenges are really the foundation of all gene cloning. So let's tackle the copying problem first. How do scientists solve it? Well, they turn to nature's most efficient little photocopiers: bacteria. The whole goal is to get a single piece of your target DNA into a bacterium and then just let it do all the hard work of making millions of copies for you. Basically, you turn these tiny organisms into microscopic factories. And this is where the genius of this whole technique really shines. See, by getting just one DNA molecule into one bacterial cell, you solve both problems at the exact same time. As that single cell divides and grows into a colony of millions of identical cells, boom, you get your amplification. And because they all came from that one original cell that had the right stuff, you also get perfect purification. Every single cell in that colony is a perfect clone, and it contains the exact DNA you wanted. But of course, there's a catch. There's always a catch. Bacteria don't just let any old piece of DNA walk in the front door. Their cell wall is like a fortress, and it's specifically designed to keep foreign stuff out. So before you can build your factory, you've got to figure out how to get past the gatekeeper. You see, in its natural state, a bacterium like E. coli has absolutely zero interest in just absorbing random DNA floating around. I mean, that's a great way to get infected by a virus, right? It's a survival mechanism. So to get our gene inside, scientists have to trick the cell. They have to make it competent, which is just a fancy way of saying they make its fortress walls temporarily leaky. And the classic method for this is honestly surprisingly simple. It's almost like a weird kitchen recipe. First, you chilled the bacteria in a calcium chloride solution. The cold slows everything down, and the calcium ions help the DNA you want to insert, which we call a plasmid, stick to the outside of the cell. Then comes the magic trick, the heat shock. You give the mixture a quick, intense blast of heat. This sudden change in temperature creates these tiny, temporary pores in the cell's membrane, giving the plasmid just enough time to sneak inside before the gate slams shut again. But here's the kicker, and it is a big one. This whole clever process, it's incredibly inefficient, like shockingly bad. Even when you do everything perfectly in the lab, it only works on about 0.01% of the cells. That is 1 in every 10,000. So if you started with 1 billion bacteria, you now have 999.9 million failures on your hands. You've just solved one problem only to create a massive new one. So with a success rate that low, your petri dish is almost entirely filled with cells that failed the test. So how on earth do you find that tiny, tiny fraction of successful cells, your needles, in that enormous haystack of failures? The solution here is just beautiful in its simplicity. You give the successful cells a superpower: antibiotic resistance. The plasmid that's carrying the gene you want also carries another gene, one that makes the bacterium completely immune to a specific antibiotic. So after you do the heat shock, you just spread all the bacteria, the millions of failures, and the few thousand successes onto a dish that's filled with an antibiotic. The antibiotic acts like a chemical weapon, killing off all the normal bacteria that didn't get a plasmid. Only the transformed cells, the ones with that resistant superpower, can survive and grow. It's a beautifully simple way to just burn away the entire haystack, leaving only your precious needles behind. But wait, it can't be that easy, can it? Of course not. There's another, more subtle problem. Sometimes the plasmid, that little circle of DNA, just snaps shut on its own, without actually picking up the gene we wanted it to carry. These empty plasmids still have the antibiotic-resistant superpower, so they survive that first test. We've got survivors, but how do we filter out these fakes from the real deal? The solution is another stroke of genius called insertional inactivation.
Think of it like this. You design the plasmid so that the only place your new gene can possibly fit is right smack in the middle of another non-essential gene. So by inserting your DNA, you literally break that other gene. And that broken gene, that's your signal. It's the flag that tells you the mission was a success. Now, the first way scientists did this was clever, but man, was it a pain. They used a plasmid that had two different antibiotic resistance genes. The target gene got inserted right into the middle of the second one, breaking it. So first, you grow everything on a plate with the first antibiotic. Then you have to carefully stamp every single colony onto a second plate with a second antibiotic. The colonies that died on that second plate, those were your winners. It worked, but it was slow, tedious, and so easy to mess up.
Yeah, this whole process of replica plating was a huge bottleneck, a total drag. Scientists knew there had to be a better, more elegant way to do this than looking for something that isn't there. They needed a direct, visible signal of success, and eventually, they found one.
A newer plasmid came along that replaced that second antibiotic gene with something much cooler, a gene called LAC-Z. When this gene is intact and working properly, the bacterial colony turns a deep blue color. But, and here's the key, if you successfully insert your piece of DNA into the plasmid, it breaks that LAC-Z gene. And the result? The colony can't make the blue color, so it stays white.
This was a complete game changer. I mean, you went from a tedious two-plate system where you had to look for a missing colony to a single plate where your successful cells are literally waving a white flag at you saying, "Pick me." The failures turn blue, the successes stay white. It was simple, it was visual, and it was fast. This blue-white screening is still a cornerstone of molecular biology to this day.
Okay, so using bacteria as tiny factories is an incredibly powerful tool. But what if you want to get DNA into other types of cells? Like yeast, plants, or even animal cells? Well, the same basic principles apply, but the delivery methods have to get a lot more sophisticated. The toolkit for this is just wild, mainly because every type of cell has its own unique defenses. For cells with really tough walls like plants and yeast, you might have to literally dissolve the wall with enzymes first. For animal cells, you can wrap the DNA in these little fat bubbles called liposomes that just merge with the cell membrane. And for a whole bunch of different types, there's something called electroporation, which is basically just a much more powerful version of heat shock that uses an electric jolt to open those pores. But of all the methods, there is one that really stands out as being particularly, direct. It's called biolistics. Yes, you heard me right. It's a gene gun. Scientists literally coat microscopic bullets of gold or tungsten with DNA and fire them at extremely high speeds directly into the target cells. It's a complete brute force method, but it's incredibly effective, especially for tough cells like in plants that resist other techniques. It's just a perfect example of scientific creativity at its most wild. So from a simple heat shock all the way to a high-tech gene gun, we've developed this incredible toolbox to find, copy, and insert DNA into living things. We've gone from just searching for a sentence in a library to now being able to rewrite the book itself. And that leaves us with the most important question of all. With the power to edit the very code of life, what problems should we choose to solve?